we actually have another iGEM friend who has been helping us with the DNA synthesis for all DNA parts that's submitted from iGEM teams. Thanks to Twist, all iGEM teams no longer have to ship their physical parts to iGEM HQ starting in 2019. I guess some of the PIs may still remember that old competition requirements, a bit stressful. Of course, DNA synthesis can be applied to many more challenges and applications. So let's welcome Emily Leproust, CEO and co-founder at Twist Bioscience, onto the main stage. Welcome, Emily.
Thank you very much. I am very, very excited to be here. I love iGEM. I think what you're doing is incredible. And at Twist, we try to be here to help you. So in my view, you are Batman, and we are Robin. And so we're really your assistant. And so obviously, DNA is very important, and DNA is changing the world. And what we do at Twist is once you have a sequence, we've built the technology to go into a tube. So we have the digital to real-life converter. And with that, there's a lot of things that can be done. And in my talk today, the first part will be about the tools that we have at Twist. And then the second part will be more about what can be done with them. But as a preview, really I feel very strongly that everything can be impacted for the better with DNA. DNA is kind of like a hammer, and everything looks like a nail. And the hundreds of iGEM teams are working in some of those great applications. So, as I mentioned, at Twist, we write DNA, and we've built a technology to really push forward the state of DNA synthesis. On the left, I'm showing how DNA and genes used to be made. People use a 96 well plate, and the chemistry of writing DNA is very well known. It's been published in 1982. It's been optimized over thousands of years of grad student. I put in my 4 years, but now you can buy a bottle of A, a bottle of C, a bottle of G, a bottle of T, and you combine them and you get any DNA sequence you want. The problem with a 96 well plate is the throughput, in my view, is too low, and the cost is too high. And so what we've done at Twist is we have developed a silicon chip, which is the same size as a 96 well plate, but instead of making 96 oligos, we can make 1 million oligos on the silicon chip. And those million oligos are clusters in 10,000 clusters of 100 oligos, and then we can do molecular biology on each of those clusters, such that we can assemble oligos into higher size DNA. If you want to get a sense of dimension, each of our million oligos is made on a dimension of 50 micron. That is the size of one hair, right? So it's extremely packed. And then we use a silicon chip to make all the orders for all the researchers at the same time on the same chip. And so we've also built a very sophisticated software infrastructure to be able to track all of that work. And so the miniaturization really gives us something that is unique, which is more scale, lower cost, and also more speed. And so to illustrate the benefit of miniaturization, we've actually engaged outside firms to measure the carbon emission that it takes to make one gene. And on the right, you can see that with a standard 96 well plate, to make one gene, it is the equivalent of driving one car for 59 miles. But with a miniaturized approach, making one gene is the equivalent of driving a car for 0.92 miles. So if you get one gene a day from Twist, you save your commute away. And it really exemplifies the power of the technology. Beyond just the miniaturization, we're also working on improving the products. And so one of the things we work very hard on is on quality. And before I get there, there's one more slide. Not only we make genes, and those genes can be non-clonal as fragments, or they can be clonal. We also make oligo pools, so there you can get from 100 oligos to 1 million oligos. That's very powerful in CRISPR experiments if you want to do genome-wide experiments to associate a phenotype with a genotype. We also can make very precise libraries for enzyme engineering or antibody engineering. And then we are also going to the pharma area, where we had a lot of customers that were getting DNA from us, but then expressing a protein, an IgG. And now we also can ship an IgG, so we'll do the expression for you. So the next few slides, I'll show some of the investments we've made, and as I mentioned earlier, the first one is in quality. In the bottom right, we are showing the % of perfect clone as you increase the length of a gene fragment. So that's a non-clonal fragment, and you can see that even at 1,800 bases, more than 50% of the clone that you pick are perfect. There's also another dimension, which is speed. A lot of what you do is around the design, build, test, learn cycle, and so the faster you can get the DNA, the faster you can get to the learning part. And on the right, you can see that this is data from our production side, where you can get gene fragments in 3 days average, and 90% of the fragments ship in 4 days. Another investment that we've made is around clonal genes. So now it's your gene in your vector. And the turnaround time for this is about 10 days. However, in the next few weeks, we'll launch an express offering where you can get a clonal gene in 5 days. So it's your gene in your vector in 5 days in a way that's very inexpensive. It's clonal perfect. And you can order one, or you can order 1,000 or 10,000. It's really, really scalable, and with very little carbon emissions. And so some of the application for that, beyond just assembling genetic circuits, we have a lot of our customers that take DNA to express IgG. And so now you can get IgG in 10 days. You've heard about RNA now being a very important new modality for therapies, and you can get to mRNA in 2 days. And as researchers develop RNA vaccines against cancer, even personalized vaccine, this is a very powerful tool. And then last, in terms of the tools that we're developing, our oligo pools used to be up to 300 base pair, which was very powerful because you could get 2 guide RNA together into an oligo. Now we are expanding our oligo pool offering, where we can go all the way to 450 base pair. And for those of you that may have synthesized your own oligos, you may be aware that the last 100 bases is the hardest part. And so it's a real big push. And with 400 base pair oligos, you're able to encode a full heavy chain or light chain in one shot. So we think that that's going to accelerate the work for antibody discovery and so on. So with that, I'll switch to our effort to support iGEM. We are very excited that last year, or this year, 120 teams used fragments and oligo pools from Twist. And we did some calculation, and over the last few months, we shipped more than 500 million bases to iGEM team, right? So it's an absolutely massive amount of DNA, and we're very, very much excited with what you have done with it. And so in the last part of the talk, I'll speak about some applications that I think could be transformative for the world, and I think you as iGEMers have had an impact, and as you go into your career, will have a huge impact. And there's really 3 big problems that I think we have to fix, and we'll fix those problems thanks to the massive amount of brain power in this room. And the first problem is really the problem of pollution. we are living a lifestyle that relies a lot on plastic, relies on fossil fuel that are extracted. We use those materials for a fleeting moment, and then they go in dumpsters and to live for a very long time. And so I think we'll talk about that, but a lot of what you do and iGEMers can impact this problem. The second problem that we have to face and that we can impact positively is the problem of hunger. And especially as climate change continues to accelerate the change, we'll see that we'll need to adapt plants to different weathers, to different pests, to different diseases. And there's a lot that we can do to ensure that the world is fed. And then the last area where iGEMers will impact is the diagnostics and the eradication of diseases. Thanks to synthetic biology and the power of the brain power of iGEMer, I think collectively we'll be able to decrease human suffering. So in the next few slides, I think I have 5 slides till the end of my talk. I just have a few questions that I'm asking the iGEMer and maybe challenge you to help with the answers. And so the first one is back to feeding the world. Can we grow food where people are? Because right now food is grown in fields, and even if we can enhance the yield, decrease the amount of fertilizer or pesticides that are being used, you still have to transport them. And as we look into the future, I think there is an opportunity to grow food where people are. And can you grow, in this case, nutritious rice or other food where people are in their apartment? And I think someone from iGEM will solve this problem. The second question is, can you adjust food, right? Can you make kale taste good? I like kale, but not everybody does. And so not only can you bring nutrition, but can you bring delight in the food that we all consume? The next question is back to pollution. I think one of the big problems is that we live in an economy that is based on plastic. And my vision is I think we can move to an economy that is based on protein. Our body is made of protein, and you can engineer protein to have whatever physical properties you want. You can have them elastic or hard or soft or transparent or flexible. And even though the last 100 years has been the century of plastic, I think the next 100 years we have an opportunity to make it the century of protein. And what that means is that it will be a world that is more sustainable, and I think it will be a world that will be also more rich in more material. And so I think the team and the people of iGEM, as they grow into their career in industry, in academia, you'll have an opportunity to make this happen, make sure that we don't need to pump oil from the ground anymore. And so to do that, we'll have to look at carbon as a system, and carbon is responsible for global climate change, but carbon is not the problem. The problem is that the carbon is in the wrong place. Carbon is in the atmosphere instead of helping us make material, helping our lifestyle, and so there's a lot of work that I think we can do to redirect and make sure that carbon is used in a circular economy. And if we do that right, I think we'll be able to lower the temperature of the planet to where it needs to be. And I think someone in this room will do that. I'll skip this slide in the interest of time. Something else that I think someone in this room is going to be, someone is going to make cancer a chronic disease. It's long overdue. We have the example with AIDS. AIDS used to be a death sentence, and now as the virus is evolving because of therapies, sequencing is used to see how the virus is evolving, and then different drugs, different cocktails are used to make sure that the virus is under control. I think the same thing can happen with cancer. Cancer doesn't have to be a death sentence. As cancer gets treated as it is recurrent, if we can develop personalized cancer treatment based on the power of synthetic biology, I think someone in this room will make cancer a chronic disease, and that will be a huge impact to humanity. In the same area of disease, there is also a big issue with rare disease, with genetic disease. Those are diseases that are quite rare, and therefore there is not a lot of investment in fixing them. But there is some amazing example. You may have heard of the recent sickle cell treatment that is a massive improvement in quality of life. And I think if we use the power of synthetic biology, we'll be able to, one by one, go and find a treatment for all of those genetic diseases. And so I think it's my last example. In disease, we've seen what happened in a pandemic with COVID, and I think through the tools of synthetic biology, we'll be able to leverage the amazing computing power and the amazing cameras that we have on our iPhone to be able to use our iPhone to diagnose and detect infectious disease faster in the future. So really, there's so many things that can be done. The world is in great need of your imagination that you've seen, that you've shown in your iGEM team. And again, as you grow and you turn into more Alec that we just heard, we can really use your imagination and what you can impact on the world. And so I really encourage you to try to find yourself amongst the greats. I'm sure that many of you will be on that picture when I come back 5, 10 years from now. I know you can do it. The world needs it, and the world deserves it. So with that, I really ask you to think about what part you will play. And when you decide that part, when you decide what kind of Robin, what kind of Batman you're going to be, then come to Twist, and we'll be your Robin to help you get there. So with that, I'll thank you very much, and looking forward to this day. Thank you.